All right, today we're diving deep into the microscopic underworld to investigate two of the most notorious bacterial families out there, the ones responsible for a huge range of human infections, staph and strep. They might be tiny, but trust me, their impact is massive. You know that feeling, that nasty sore throat, a really painful skin boil, or a sudden awful bout of food poisoning? Well, chances are you've already had a run-in with one of our main culprits. They're totally invisible to the naked eye, but you definitely feel their effects. So, let's meet our prime suspects. On one side, you've got staphylococci. They like to hang out in these tight grape-like clusters. On the other side, you have streptococci, and they form these really distinct chains or pairs. But you know the first real clue that tells them apart? It's something called the catalase test. Think of it like this. Staph has a special enzyme that lets it break down hydrogen peroxide. Strep doesn't. So, staph is catalase positive, strep is catalase negative. It's a simple test, but it's the first huge step in cracking the case. Okay, let's start our profiling with family number 1, the Staphylococci, or as we're going to call them, the grape-like gang. And topping the most wanted list for this family is its infamous ringleader, Staphylococcus aureus. This is the bad guy behind some of the most common and, frankly, some of the most dangerous bacterial infections that we have to deal with. So let's pull up its case file. Its number 1 distinguishing mark is that it's coagulase positive. Now, coagulase is the sneaky enzyme that clots blood plasma, basically creating a perfect little shield for the bacteria to hide from our immune system. Its MO? Oh, it's everything from skin infections to really life-threatening stuff like toxic shock syndrome. And it pulls this off with a whole arsenal of secret weapons, like hyaluronidase, a spreading factor that literally dissolves the glue holding our cells together, letting it invade deeper into our tissues. And one of its classic moves? Food poisoning. When you get food, think potato salad, cream pastries, contaminated with its preformed toxin, the results are incredibly fast. We're talking symptoms hitting you like a ton of bricks, usually within just 2 to 6 hours. Now, this is where S. aureus goes from a common criminal to a modern-day supervillain. A lot of strains have figured out how to defeat our best drugs. This is how we get superbugs like MRSA, that's methicillin-resistant S. aureus, and they are so hard to treat. For these really tough cases, doctors have to bring out the big guns, often a powerful antibiotic called vancomycin. But hey, the big boss isn't the only member of the gang, right? You've also got the opportunists, the coagulase-negative staphs. They're usually not as aggressive, but they can cause huge problems, especially for people who are already sick or have medical devices like catheters or artificial joints. So how do we identify these accomplices? Well, the lab uses a different clue. The novobiocene test. Staphylococcus epidermidis, which is notorious for causing infections on prosthetic devices, is sensitive to this antibiotic. But its cousin, Staphylococcus saprophyticus, a big-time culprit in UTIs, is resistant. It's another simple test, but it's all it takes to point the finger at the right suspect. All right, time to switch our investigation over to the second major crime family, the Streptococci. We call them the chain gang, and you can probably guess why based on how they look under the microscope. So, what's their deal? Well, they form those classic chains or pairs. And remember that key difference from staph? They are all catalase negative. They're also sorted into something called Lancefield groups, which is basically just a way of categorizing them based on a specific molecule on their cell surface.
And just like any good crime family, the Streptococci have some real notorious members, the all-stars responsible for a whole host of human diseases. Let's run down the lineup.
This is where the lab work gets really clever. Almost like bacterial fingerprinting. They can pinpoint 2 of the biggest culprits using 2 different antibiotic discs, bacitracin and opticin. Streptococcus pyones, the bug behind strep throat, is sensitive to bacitracin, but Streptococcus pneumonia, a huge cause of pneumonia, is sensitive to opticin. It's a clear-cut way to get a positive ID. Let's focus on S. pyogenes for a sec. This is the one behind strep throat and skin infections like impetigo. But the real danger isn't always the infection itself. It's what can happen afterwards. It can actually trigger an autoimmune reaction, leading to really serious conditions like rheumatic fever, which can permanently damage the heart. Now, for its cousin, S. pneumoniae, knowing what medicine to use is everything. So, labs will test how vulnerable it is to penicillin by measuring the zone of inhibition. That's the clear area around an antibiotic disc where the bacteria just can't grow. If that zone is 20 millimeters or bigger, that's a great sign. It's a clear signal that penicillin is going to work. Next up in the lineup is Streptococcus agalactiae, or Group B strep. And this one is especially dangerous, a true nemesis for newborns. It's a primary cause of life-threatening neonatal sepsis and meningitis. And finally, we've got the gut gang. This includes Group D strep and a really tough character called Enterococcus. That entero part of the name is a big hint about where it lives, the gastrointestinal tract. So how do you single out the incredibly tough Enterococcus? It's a clever two-step process. First, both Group D strep and Enterococcus can pass something called the bile esculin test. They turn the test media black. But the second step is the real kicker. Only Enterococcus is tough enough to grow in a really high salt environment. If it can grow in 6.5% salt, boom, you've found your culprit. And this is exactly why telling them apart is so critical. Finding Enterococcus is often linked to UTIs or even heart infections, but finding its relative, S. bovis, in a person's bloodstream, that can be a massive red flag for something else entirely, colorectal cancer. So we've gone through all this detective work, from catalase tests to salt tolerance. Why? Why does correctly identifying these microscopic culprits matter so much? Well, the final verdict couldn't be clearer. Accurate identification is literally the cornerstone of modern medicine. It makes sure patients get the right treatment instead of the wrong one. It guides doctors to choose the most effective antibiotic. It helps us track and hopefully stop the spread of superbugs like MRSA. And sometimes, like we saw with S. bovis, it can even provide life-saving clues to other major diseases. From those grape-like clusters to those long chains, this microscopic underworld is just packed with clues. And learning how to read them correctly saves lives every single day. The thing is, these organisms are always evolving, always developing new defenses. So the real question is, in this ongoing fight against infection, what's going to be our next clue?